My name is Varanya, and I want to share with you a little bit about our next phase as well as about the GenRe-Mekong project. So as we approach the last milestone in malaria elimination control program, in the malaria elimination, control programs have to face new challenges, such as prioritizing elimination-focused use cases, navigating shifts in the epidemiological landscape, and managing the emergence of non-falciparum species. The GenRe-Mekong is also transitioning to a new phase to support these new challenges. So in this talk, I will focus on two use cases to support elimination goals. One is the identification of imported cases, and the second is the characterization of outbreaks. But before I do that, I want to start by touching on the phase 1 and phase 2 of the GenRe-Mekong project and what the project has achieved so far. The primary goal of GenRe-Mekong is to support national malaria control programs in their decision-making for malaria control and elimination by providing relevant, timely, and comprehensive actionable knowledge about the genetics of the circulating parasites population. And to achieve this goal, in phase 1, GenRe-Mekong has established technical platforms, processes, and analysis techniques to enable large-scale genetic epidemiology. We have processed over 13,000 PF samples and have successfully established regional labs. GenRe-Mekong has built direct, strong partnership with the control programs in 4 GMS countries, in Vietnam, Laos, Cambodia, and Thailand. And it is our pleasure and honor that all of you are here with us in this room today. And through the control programs network, dry blood spot samples of malaria-positive patients are collected and sent to one of our regional labs for genotyping using amplicon sequencing. We then analyze the genetic data in a way that is relevant to national malaria control program activities and communicate these results back to the control programs. In our Spot Malaria platform, we genotype many markers that have been validated to be associated with anti-malaria resistance. These are used for the resistance prediction. We also genotype 100 variants across the genome called the genetic barcodes, which are like a summary of the genome, and we use these to compare between a pair of parasites to see how similar they are, how connected they are. So in the second phase of GenRe-Mekong, we focuses on supporting NMCP with actionable insights such as tracking resistant strains, monitoring the spread of resistance, and estimating drug efficacy. During the period in which DHA piperacin resistance spread across the region, insights from genetic surveillance has been used by the control program to support the decisions on frontline therapy choices. For example, genetic surveillance results has supported Vietnam in categorizing Tier 1 provinces to switch away from using DHA piperacin as frontline therapy in these high resistance provinces. In a similar way, the surveillance result has supported Laos during the review of frontline therapy, showing that it was not a good idea to adopt DHA piperacin during this period because of the spread of resistance of parasites in the lower zone of southern Laos. It is important to continue monitoring resistance post-guideline transition to provide control programs on the impact of interventions and provide early warning of any developing resistance. And molecular surveillance can cover a broad range of drugs beside the first-line therapy, and therefore these outputs can support evaluation of alternative therapies in addition to monitoring the predicted efficacy of the current frontline. So in the past few years, the region has seen a rapid decline in resistance to piperacin. since the regional switch away from DHA peppercorn usage, as well as we also see a rapid decline in the PF malaria cases. And this decline in PF prevalence was particularly observed in the eastern part of the GMS. And so it seems we are approaching the last milestone in malaria elimination. And with this shift in epidemiological changes, the control programs are facing new challenges and have different technical needs. And so our aim for this forum is to start talking about these priority use cases to support the elimination goals. GenRe-Mekong will continue to monitor the genetic markers and tracking of resistant strains because these are important. And we will be developing tools to empower control program and the research community to be able to do genetic epidemiology analysis. One of such tools that we have developed is the web application. We have recently developed this for quick exploration and visualization of the GRC data. And so we will have a short workshop on this in the afternoon. Will be given by Dr. Vipavi from the GenRe team. Being able to identify imported cases and prevention of reintroduction is important for achieving malaria-free certificates. So we have Professor Du Quan from the National Institute of Parasitic Diseases, China CDC, who will be sharing with us this morning lessons from China malaria elimination. In addition to identifying imported cases, outbreak will become more common as we approach elimination. And so I will be providing information of how GenRe-Mekong can support these use cases today. Another new challenge is the emergence of non-falciparum species. Originally, we have Professor Malika, who would be giving us a talk on this, but unfortunately, a situation has arisen, so she cannot make it today. She sends her apologies. But if you are interested in learning about the methods that her team has developed, which is a hexaplex PCR method that is simple, low cost, highly sensitive for detecting 5 human malaria species, you are very welcome to check out her work as shown on the screen. And excitedly, we have started and made progress in the research and surveillance of Plasmodium vivax. And so we have Professor Sarah and Anto from Menzies School of Health Research, who will be talking about the molecular tools for vivax and predicting their geographical origins. We also have Ethan Booth from the GenRe-Mekong team, who will share with you our initial hot off the press vivax surveillance results. And all of these talks will be just before lunchtime. And yes, we know that there is a great interest in vector surveillance for malaria control in this region. And so we have Professor Martin from Liverpool School of Tropical Medicine, who will be sharing with us his expertise on this use case this afternoon. And finally, in the last session, we have Professor Olivo from GenRe-Mekong team, who will be exploring with you the different technologies to support surveillance depending on the use case or what answer you're looking for. So all of these exciting talks to come. But for now, I would like us to shift our mind to the importation use case. To apply for WHO malaria-free certification, countries have to demonstrate that all of the malaria cases are coming from abroad and no indigenous cases for at least 3 years. When there are cases, control programs may be asking, are these cases imported? If so, where do they come from? The typical method that is used right now is to do a travel survey, which is quick and simple to do. But the downside is the recall biases. Like the information depends quite a lot on the patient's ability to recall their travel history and their willingness to share about where they have been to. There is also uncertainty in the infection origin. Another method that some research group has been investigating is through the use of GPS tracking, which can measure spatial spread, but very limited logistically and difficult to implement practically. So for us, GenRe-Mekong aim to support control program in addressing this question by offering an additional tools, a molecular genetic approach in which we compare the genetic composition of the parasites across different regions. So we can compare the parasites by estimating the proportion of SNP where the alleles differ. And we can do this using a few SNPs or haplotype. In our current platform that we are using, we already been using 100 SNP barcodes to compare how similar a pair of parasites are. One can also use whole genome sequencing, which will give us hundreds and thousands of SNPs. Having more information, like having more number of informative SNPs in general, will provide higher resolution. And depending on the complexity of the problem, lower resolution genotype can already answer some of these questions. If, for example, the importation is coming from countries that are far apart, it is possible to just use a few SNPs to distinguish the parasites. But when we try to distinguish parasites from neighboring countries, we may need to use higher resolution, like the genetic barcode or even whole genome sequencing. The 100 SNP barcode in our GRC of the current platform is able to answer many of these cases between neighboring countries already, which I will show you in a second. But however, in this region, there is a problem with the spread of resistant strain, which has been spread across the region. And this makes it defining the origin of these parasites that belong to these clusters a lot harder. Okay, so what can we do today? Without changing our current genotyping platform, we can use the 100 SNP barcode to distinguish African and Asian parasites very well. If the parasites are coming from any countries in Africa and into GMS, we can train a machine learning algorithm to predict importation with extreme accuracy at 99%. For identifying samples between Laos, Vietnam, and Cambodia, we can use another machine learning algorithm to predict origin. Though the accuracy reduces, we can still predict origin with high accuracy at 81%. So why is it hard to distinguish parasites between Laos, Cambodia, and Vietnam? What makes it hard to pinpoint the origin of the parasites is because of the high level of cross-border traffic between these countries and the highly similar genomes and the spread of the resistant strain that goes into all 4 countries of the GMS. And for these strains that spread into several neighboring countries, what we can do is we can classify them into clusters. So if the parasites belongs to one of these clusters, we can then give you a lot of information about them, like where they are present and about their drug profile. So for example, with today's GRC barcode, if a sample is collected in Laos and the question is whether they have been imported or not, the first question that we ask is, does this belong to a multi-country cluster? If yes, then we can provide details about the cluster where they circulate and also about their drug-resistant profile. But if not, then we can provide the country of origin prediction with accuracy probability. For example, like origin Congo, accuracy 98%, imported yes. In the future, we plan to improve on the analysis methods to be able to predict the origin even better, even if they belong to one of these clusters. But today, this is what is available. The control program can use genetic data to complement and support the travel data to help with the decision making on importation. And in the future, if we adopt whole genome sequencing, this would help to enhance resolution and improve accuracy of predictions between neighboring countries. Whole genome sequencing will also enable detailed analysis of the parasite ancestry and help us to have a deeper understanding of the population, genetics, and the gene flow. Okay, next, I'll be sharing with you how genetic epidemiology can be used to support outbreak characterization. As we are approaching elimination, outbreaks will occur more often and in unpredictable places. So once control program has declared that there is an outbreak, they might be wondering, what is the driving cause for the outbreak? Was it because of the heavy rain, more people going to the forest, more mosquito or resistant parasites? So these are all important questions, because being able to distinguish what is the driving cause of an outbreak will help control program to decide on the most effective response. Again, the typical assessment that is used currently is to use clinical and travel data. And GenRe would like to support the current method by offering the genetic epidemiology approach, assessments of outbreak. Analyzing the genetics of parasites that is causing an outbreak can inform us to a certain extent about what might be the driving cause. This is because we expect different genetic signature for outbreaks that are driven by genetic genetic selection versus an outbreak that are driven by increased vector activities. In particular, we expect to see different population structure and diversity between these type of outbreaks, as I will explain to you in the next few slides. In addition to assessing the diversity and population structure, what we can also do is we can monitor genetic markers to assess the spread of resistant alleles. We can identify circulating strains and track their origin. And these are all possible because we have built large data sets in phase 1 and phase 2, in which we can then compare the outbreak strain to what was circulating before that outbreak to gain more insights about where they might be coming from and whether they are locally transmitted. So, under normal circumstances with no selective pressure, malaria parasites are expected to mate randomly, undergoing sexual recombination in mosquitoes that control the diversity within a population. However, when the epidemiological events occur, this can cause a departure from the expected patterns. For example, strong selection of a drug-resistant strain makes it more likely that the members of this strain would be mating with each other, and then we would have a lot of the clonal identical strains expanding, and a loss of diversity in the population. So these patterns we may be able to detect them even without any knowledge of what is the driving cause, without knowing which gene was responsible. So this gives us an early warning sign that we can go and investigate further that there's something going on. Genetic selection reduces population diversity because parasites with advantageous mutation multiply and thrive. And so an outbreak driven by selection might be characterized by a significant reduction in population diversity and expansion of large clonal clusters. On the other hand, we expect to see a different genetic profile if the outbreak is driven by increased vector activity or increased human movement. In low transmission setting, like the Greater Mekong Subregion, a person is usually infected with one parasite type. When there is no selection or preference towards one type, and there has been an increase in vector activity, we are expected to see an increase in number of cases, but then similar level of the parasite diversity compared to the period before the outbreak. Increased vector activity may also resulted in the increased parasite population diversity because of the increased chances of outcrossing, which will result in a recombinant offspring. And so as long as there was diversity to begin with, we expect an outbreak that are driven by increased vector activity to maintain or increase the diversity pattern. To help support outbreak characterization, what we can do today is that we can assess diversity and the population structure to identify whether an outbreak is caused by a clonal expansion. We can assess genetic markers and identify the circulating strain. We can also analyze whether it is a local strain to that province or assess whether it might be coming from somewhere else within the country or from outside of the country. So I hope that I have shown you today that genetic epidemiology can support last mile use cases, and we plan to develop the methodologies and tools to be able to address more questions. However, if you look at the bit with the plan, there are several areas that I have not talked about, but it is as part of the plan for the next phase, and these we will explore together as part of the forum discussion. But before I finish, I would like to highlight that faster turnaround time is needed to support all of these last milestone use cases. So in phase 2, the control programs predicted and shows the location for sample collection, and then samples are then accumulated at the sites before sending to one of our regional labs. Current turnaround time is around 8 months. However, situation now required a different approach and a faster turnaround time is needed. So now we have minimal number of cases and that is emerging randomly, and we can no longer predict where the outbreak might be or where the imported samples may be sampled from. And so we will be exploring this topic together later today. But the questions that I want to leave us to start thinking about is, what can we do together as a group to respond more quickly? How can we give answers more quickly? Like, how can the samples be picked up in the most efficient way? And with that, I would like to thank our funder at the Bill and Melinda Gates Foundation and the Global Fund. And thank you to all of the control programs partners and our scientific partners. And thank you so much for listening to me for such a long time. Thank you. And is there any questions?
Any questions for Varanya, please? Yeah. Dr. Siv, you can use the microphone there. Okay.
Yeah, thank you very much for a good presentation. There is will be important for imported case, very interesting for our country. I just brief you that currently Cambodia, 90% of PM malaria is imported. We identify some of more of them import from Central Africa. Yeah, because some people go to work and come back with PM malaria. 90% right now this year. My question is, you has been talk a lot about the title of the last mile, but it's not really a last mile. You have to say that how to prevent or re-establishment of malaria. Yeah, because you talking about imported case.
Yeah.
Yeah. And also one technical question, but I'm not clearly captured that. But you said that in order to identify whatever is the origin of the outbreak of that, you can do the genetic epidemiology to detect and also link to the original pattern of the marker that already existed. So if there is a look like, it seems to be that it's the same strain or something like that. But my question is, how far of one generation to the other generation? Because of the original strain, will become out with the mosquito with the same strain. But how long they can switch to the other? That's you can detect it.
Sure. Thank you so much for your question, Dr. Siv. Actually, we have observed that in the previous outbreak investigation that we did observe that some strain have been circulating in the region without any changes at all in the last 10 years. That has been almost identical in that genome. And then whereas other strains, we also do see, with using the genetic barcodes, we already get a sense that they are a descendants of like a certain strain. But then when we use the whole genome sequencing, we were able to see which region of the parasites are inherited from which ancestry. Given that we have samples, it really depends on the database that we have as well. We can only go back as far as we have something to compare to, in the past.
So, if I could just add to that, I mean, we're very surprised how little some of the drug-resistant strains have changed over time. Probably because they have like multiple mutations that they must carry, and if they recombine with others, they may lose the sort of magic formula, as it were, right? So it is quite surprising, and we also could see quite clearly the patterns whereby some different strains are recombined together. Now, that was sort of until the changes in ACT frontline policy. It could well be that now that different drugs are being used, we may see a more substantial change in the lineage. It's not clear because the numbers have gone down so much. It's very difficult to study right now. Yeah.
Thank you.
Yeah, maybe
That's okay. Yeah, thanks. Really nice presentation. Just a couple of extra comments, which might come up later on as well, is in addition to POR, where I think if we're having cases coming from other countries, often the travel history might be a bit more accurate, is thinking a bit about subnational verification as well, where it might be a bit more challenging to say whether cases have come from a different province or different district. But I think that's probably also thinking about the first step of POR, is that countries might want to do subnational verification. So thinking, okay, did they bring the parasite from another province within the country, and that would affect the subnational verification for the province they're in. The other thing that might be interesting to look at is whether we can link their travel histories or their movement data, if you're tracking it through mobile phones, with some of the genomics as well. I don't know if that work's currently being done, but that might be another nice thing to also validate some of this work.
Yeah, we have done some of that in the past, and in fact, I think perhaps Cambodia is still collecting, right? Yeah. And we do collect this information. Whether or not the stories we're getting are reliable, that has depended a lot on what type of activities people were conducting. But yeah.